Hi, everyone. Good Tuesday to you. Welcome to our Tuesday seminars on the cerebellum. Today, we're going to hear from a young postdoctoral fellow, Robyn Mildren. Robyn received her PhD in kinesiology from University of British Columbia, where she worked on the human motor control, studying the ankle and leg muscles and the neurophysiology of response to tactile and vibratory stimuli. Then she joined Kathy Cullen's lab here at Johns Hopkins, where she is a Kavli Distinguished Postdoctoral Fellow. She's been studying non-human primates, looking at the cerebellum and its role in the control of self-motion vestibular inputs. So, Robyn, it's good to have you with us. And the other thing important about Robyn is that she's absolutely an elite athlete. She not only studies motor control, she performs at very high levels. I'll just give you a little bit about her performance. She finished top 3 in the Vancouver's marathon in 2019, and she was a 3rd Canadian in the Houston Marathon, also in the 2019. So she's an amazing runner. Look forward to your talk, Robyn.
Thanks for that introduction, Reza, and also thank you so much for giving me the opportunity to give this talk. So I'm going to be talking about predictive processing of self-motion by the primate vestibular system. And so, as you all know, the cerebellum is essential for coordinated movement and balance. And it generates internal models of our self-motion and our spatial orientation relative to gravity. And it does this by integrating vestibular along with other sensorimotor information. And so you're all already experts in the cerebellum, but I'm going to provide a little bit more background on the vestibular system. So the vestibular system is comprised of these 5 sensory organs. We have the 3 semicircular canals, which provide information about angular motion of the head, and 2 otolith organs, the utricle and saccule, which sense linear accelerations. And so together they can provide information about the motion of the head in space as we go about our activities of our everyday lives. And they transmit these signals through the 8th cranial nerve to central vestibular neurons in the brainstem and the cerebellum. And via these pathways, the vestibular system contributes to 3 main functions. One is stabilizing gaze via the vestibulo-ocular reflex. Another is stabilizing posture via vestibulospinal reflex. And then finally, self-motion perception and navigation. And so, although the vestibular system contributes to these 3 functions, I'm mainly going to focus on my main interest, which is postural control. So one important question that the brain needs to answer is, did I mean to do that? In other words, is the sensory information that the brain is receiving a result of its own motor actions or from some unexpected perturbation? And that's an important question to ask because, for example, in this scenario, this figure skater is making this jump. And so the head acceleration that she's experiencing is going to, if it generates vestibulospinal reflexes, it might be counterproductive to her movement goals. In contrast, in this situation, this individual is slipping on a slippery floor. And in this case, he will want robust vestibulospinal reflex to help adjust his posture or correct his balance. So prior work by Dr. Cullen has looked at how the brain distinguishes between active versus passive self-motion. So here they recorded from the first central processing stage of vestibular information, the vestibular nuclei in the brainstem. And this is in rhesus monkeys while they're on this vestibular turntable where they can do yaw angular motion. And so this is just showing the head in space velocity, the active component, which there is none in this situation. It's entirely passively generated. And then the passive velocity of the turntable, and the shaded area is the firing rate of this vestibular nuclei neuron. And you can see it really well tracks the motion of that platform. However, when they let the monkey do an active voluntary head movement for a reward, this is the head in space velocity. It's generated by the active movement. There's no passive component. And the shaded area shows that this neuron's response is markedly suppressed relative to what you would predict based on its response to passive self-motion. And this final condition is really crucial because it shows that there is a precise prediction and suppression of the self-generated self-motion signals. So here, as the monkey goes to make a head movement, they actually counter-rotate the turntable such that the head remains nearly stable in space. So this is the active component, this is the passive component, and the neuron firing actually tracks the passive component of that motion, despite the head staying nearly stable in space. So overall, the model we have is that the brain generates a forward internal model of the sensory consequences of our motor commands and compares that with actual sensory feedback. And if there is a match between predicted and actual feedback, the vestibular reafferent information is canceled at the level of the vestibular nuclei, thus not leading to postural reflexes. However, if there's a mismatch between predicted and actual feedback, the vestibular input is not suppressed, leading to robust vestibulospinal reflexes to help correct our posture. So similar things have also been shown in the deep cerebellar nuclei, specifically the rostral fastigial, which is the most medial one that receives or processes vestibular information. And so again, this is plotting head velocity during passive motion. This neuron nicely follows that. During the active condition, the neuron firing is markedly suppressed, but here they apply a velocity-dependent torque to the monkey's head as he makes an active head movement, such that it reduces the head velocity to about half of what the monkey intended to do. And you can see when there's this sensory prediction error, the neuron responds as if that motion was passively generated. And this is just plotting the head velocity and the sensitivity of the neuron, passive, active condition, and during sensory prediction error. But over time, as they leave that velocity-dependent torque on, after you get to about close to 50 trials, the monkey learns this new relationship between the motor commands he produces and the resulting sensory feedback that's going to be received. And so as his head velocity increases back to normal levels, the firing of that neuron goes back to encoding that motion as if it were a normal actively generated movement. And importantly, in catch trials, if they suddenly release that velocity-dependent torque, the head movement is much faster than the monkey expects, and you again see that treated as if it was an external perturbation. And so overall, neurons in the deep cerebellar nuclei, which are the output of the cerebellum, they tend to show the outcome of this comparison between predicted and actual sensory feedback. And so this kind of leads to the first question, which is, how is this computation generated? And for this, the area that we're interested in looking at is the anterior vermis Purkinje cells, which project both to the deep cerebellar nuclei as well as the vestibular nuclei. And so first we want to look at how self-motion is represented in these anterior vermis Purkinje cells. And then next, how do they process predictable self-motion information? So it's first important to consider that the vestibular system needs to take into account the position of the head relative to the body in order to generate appropriate vestibulospinal reflexes. And we know that that is the case based on recordings from deep cerebellar nuclei, where there's two distinct groups of neurons. First, there's these unimodal neurons, which provide information about motion of the head in space. So they'll respond if you rotate the monkey's whole body, or if you rotate just the head on the body, but not if you rotate the body underneath the head. They're not tracking body motion, they're tracking purely motion of the head. This other group is bimodal neurons, and they track motion of the body. So if you rotate the monkey's whole body, they nicely modulate with that. If you actually keep the head stable in space and move the body underneath the head, they use that proprioceptive information to signal motion of the body. And then if you move just the head on top of the body, there's no motion of the body and they do not respond. So we know there are these very distinct classes in the deep cerebellar nuclei. So the question is, how might that be computed by the anterior vermis Purkinje cells that project to those neurons? And so the work that I'm going to be talking about was recorded by Omid Zobiri. And so first he looked at how these Purkinje cells in the anterior vermis respond to passive whole body motion, which is a vestibular stimuli. So this is showing one example Purkinje cell. This is the velocity of the head and body, which is the same, and the mean firing rate of the neuron, as well as the firing across trials. And you can see that this Purkinje cell nicely shows excitatory responses in one direction, inhibitory responses in the contralateral or the ipsilateral direction. And so he called this a linear Purkinje cell. But interestingly, he found a lot of other types of Purkinje cells as well. Some respond with this more V-shaped pattern where they have excitatory responses in both directions, or some he termed rectifying, which show responses mainly in one direction. And the findings were similar with looking at the responses to neck proprioception in the condition where you hold the head stable in space and move the body underneath the head. And so again, there's some V-shaped, some rectifying, and some linear neurons. So overall, if you plot these two populations of neurons in the deep cerebellar nuclei, we have again our unimodal neurons that have a head sensitivity ratio of 1. They're only encoding head motion. The bimodal neurons who have a body sensitivity ratio of 1, a head sensitivity ratio of 0. If you plot the Purkinje cells on top of that, you can see that a lot of them fall somewhere in between this head and body coding space. But interestingly, due to the heterogeneity in their response and the heterogeneity in their dynamics, if you do a simple linear weighted combination of a population of Purkinje cells, you can start to predict the responses of the target RFN neurons. And that prediction does pretty well when you get to a number of around 40 Purkinje cells, which is interesting because that happens to be the natural convergence ratio of Purkinje cells onto the RFN neurons. So next you want to address the question, did I mean to do that? Or how do these anterior vermis Purkinje cells potentially contribute to this prediction and suppression of self-generated vestibular feedback? And so first, it's important to note that the primary vestibular afferents coming from the sensory organs do not distinguish between active versus passive motion. And that's been shown even in very complicated scenarios like walking and running, where they follow the head motion just as well as if it was passively applied. Yet, as I said before, when you go to the next stage of processing in the vestibular nuclei, also in the deep cerebellar nuclei, there's marked suppression of the cell responses to active self-motion. And so first he looked at responses to passive compared to active head motion. And you can see, somewhat surprisingly, the Purkinje cells also showed inhibitory response or suppressed responses to active head movements relative to comparable passive movements. And that's perhaps surprising because Purkinje cells are inhibitory onto their target neurons. So if they were generating that suppression, you might have expected them to even have enhanced sensitivity during active movement. And they also did show responses to the pure motor command. So when the monkey went to make a head movement and they unexpectedly blocked that movement such that it wasn't realized, there was some modulation in the neural activity. And combining the passive condition along with the motor command condition does a fairly good job of predicting how these neurons respond during active head-on body movements. And so, interestingly, despite the suppressed responses during the active condition in the Purkinje cells, again, due to the heterogeneity and the diversity of the way that they respond to different sensory motor signals. Again, pooling the population activity, doing a weighted linear combination can begin to predict the responses of their target RFN neurons during the active condition. So overall, this work shows that the anterior vermis plays two key roles. One is transforming head-centered vestibular signals into a body-centered reference frame, which is crucial for generating appropriate vestibular spinal reflexes. And the other is predicting and canceling the sensory consequences of our active self-motion. So the majority of previous work in the lab has addressed very large perturbations or externally imposed self-motion or deviations from intended head movements. What we wanted to address next is what happens when you just make small errors that are even within the natural level of variability of movement. And so instead of doing these abrupt, large velocity-dependent torques, we applied subtle different levels of torques that changed the intended head velocity just outside or even just within the natural range of variability. And you wanted to ask, how do rostrovestibular neurons respond to these small perturbations that only subtly influence the intended movement? And we had two hypotheses. One is that you would see this abrupt change in tuning as soon as you get outside the natural amount of movement variability. The second hypothesis is that you would see this gradual change in sensitivity of the neurons as you go from your regular active-like movement towards larger perturbations where you would see more passive-like sensitivity. We also wanted to look at how the cerebellar Purkinje cells generate responses in these target neurons under these conditions and how they might respond to the sensory prediction error. So in this study, we had the monkey making active head movements for a reward. And in a very small fraction of the trials, we applied one of 4 levels of velocity-dependent torques. And they were carefully titrated such that some of them created just very, very small deviations in the intended head movement speed. And importantly, we went in both directions. So we had some torques that assisted the head movement, so made it a little bit faster than the monkey intended, and some that resisted the head movement, so made them a little bit slower. And during this, we recorded from 3 areas. First, the RFN neurons. We also recorded from anterior vermis Purkinje cells, and then finally 2 neck muscles, sternocleidomastoid and splenius capitis, in order to look at the functional outcome of these perturbations. So for the recordings in the cerebellum, we use these 128 channel silicon read-write probes. They're very similar to neuropixels, although they're passive. You can actually both stimulate and record through all of the channels. And so I'm just going to share with you, I can't remember if I shared audio. I'm going to share one example recording. Let me know if you can't hear it. No? Now I share screen again and remember to share audio. Okay. Share sound. Okay. Sorry about that. I need to write a sticky note to remember to do this. Okay, could you hear it that time? Okay, awesome. So what you're looking for is there's this sinusoidal motion of the turntable. This is just a passive angular yaw rotation. And hopefully you can hear the modulation of that RFN neuron as you do these passive motions. Okay, so similar to previous work, when we recorded from these RFN neurons during passive motion, we see this beautiful modulation in the neural activity. However, in the active condition where the monkey makes an active head movement for a reward, there's markedly suppressed sensitivity. And so next we want to look at what happens when you introduce small perturbations that only subtly influence the intended head movement. And so we went in both directions, assistive and resistive. And here are some of the results. So we subtly influence the head velocity, either making it a little faster or a little slower than the monkey intended. And these are the responses seen in the RFN neurons. So surprisingly, even with the very smallest perturbation levels, the sensitivity of the neurons markedly changed from active-like levels, completely suppressed, up to passive levels. And that was true in both assistive and resistive directions. So next we want to look at the Purkinje cells, which provide input to these RFN neurons. And we saw something different, actually. They showed this more gradual adjustment in their sensitivity from suppressed levels during normal active head movements, approaching passive-like levels, as if the movement was entirely externally generated. And so interesting, again, if you start to pool the responses of a population of Purkinje cells, once you get to about 30 Purkinje cells, you can do a fairly good job of predicting the responses in their target RFN neurons. And so ultimately, if we compare this back to our hypotheses, they look a little bit more like hypothesis 2, but the RFN neurons show this much more abrupt adjustment in their sensitivity, even within natural range of movement variability, whereas the Purkinje cells show more of this gradual change. And then finally, when we looked at our EMG responses, they were particularly prominent in the assistive direction when you record from the contralateral muscle. And you could see that even with small perturbations, there are some vestibulospinal reflexes generated in that muscle that help to stabilize the head in space, such that if the head movement is a little bit faster than intended, the right splenius capitis muscle, if the movement is in the left direction, would be activated in order to help stabilize the head in space. Okay. So moving on to the final part of this talk, I'm going to talk about how self-motion is represented in the posterior vermis, specifically in the nodulus and uvula. And this is a really interesting area to study because it's a real hub of processing vestibular information. And so it's known that the nodulus uvula integrates both canal and otolith inputs in order to form a representation of our orientation and self-motion relative to gravity. And this sense of spatial orientation is really essential for stabilizing both gaze and posture. So for example, lesions of the nodulus uvula generate impairments in the ability to align the VOR with gravity. It's a really interesting area to study too, because it's the only region of the cerebellum that receives direct primary vestibular afferent input via mossy fibers. And so we might suspect that it might process active self-motion in a different way compared to the rest of the cerebellum. So in this study, again, we use our read-write probe to record the activity of neurons in the nodulus and uvula while we applied these 3 different passive stimuli conditions. And here we looked at translations in both the anteroposterior and medial-lateral directions. So first, we applied passive whole body translations, where we're translating the whole monkey in space. And then proprioceptive stimulation, where we actually held the monkey's head stable and translated the body underneath the head just to stretch neck muscle spindles. And then finally, in this combined condition, we move the head on top of the body in order to stimulate both the vestibular system and neck muscle spindles. And then finally, we release the head sled and allow the monkey to make active head movements for a reward, and compared that to the passive head-on body condition. So this is showing an example response of a Purkinje cell in nodulus and uvula during the two passive stimulation conditions. So we found that this is plotting the head velocity, mean firing rate of the neuron, and then the firing across different trials of motion. And so, as you can see, we found that most of these neurons actually respond to both vestibular and proprioceptive stimulation. And then when we did the combined condition, which was the head-on body, they also showed some nice responses encoding that head motion. And so if we looked at combining both the vestibular and the proprioceptive condition in order to try and predict responses to the combined condition, which is head-on body, we showed that we could do a pretty good job if you look at the sensitivity to head-on body translations compared to that predicted based on summing, doing a vector sum of the responses to vestibular and proprioceptive. They're fairly well matched in terms of both their sensitivity as well as the phase in both the AP and ML directions. We also looked at some directional tuning. So for example, this neuron showed responses in both the AP and the ML directions. And so you can calculate its estimated maximum tuning direction, which is somewhere in between AP and ML. If I bind all the responses of all the Purkinje cells, there's a little bit of clustering towards pure AP, but for the most part, these neurons fall within that whole spread between AP and ML tuning. And then finally, interestingly, we want to look at how they respond to active self-motion. And surprisingly, or interestingly, compared to all the other areas of the cerebellum that we've previously looked, this area did not distinguish between active versus passive self-motion. And there was no significant suppression during active motion relative to similar passive motions. We also did not see significant responses to the motor command condition, where we unexpectedly blocked the monkey's head movement such that motor commands were generated, but there was no motion of the head. So overall, to summarize, the posterior vermis, we find that these nodulus uvular Purkinje cells combine vestibular and neck proprioceptive inputs in order to form an estimate of head motion. And these Purkinje cells show no significant suppression of vestibular reafference, and this is in contrast to other areas of the brain, including the anterior vermis, both the deep cerebellar nuclei and the vestibular nuclei. So in this final part of the talk, I want to talk about some ongoing work where we're trying to probe the vestibular spinal reflexes and the pathways involved in those. And so we've been recording EMG from neck muscles, and this was done with just fine wire EMG and applying sinusoidal motion to the monkey as a vestibular stimulation. And we've tried to parse apart the influences of the vestibular system and other sensory inputs as well, including manipulating visual information. And you can see during sinusoidal motion, there's nice modulation in neck motor units in normal monkeys, not in vestibular loss monkeys. And so since the majority of motion we experience is never really a pure sine wave, we also looked at responses to broadband motion that contain frequencies up to around 20 hertz, which is important because when Kathy has previously put sensors on individuals and also on monkeys and track them as they go about their daily activities, you do see motion containing frequencies up to around 20 hertz. And so we do see coherence between motor units and self-motion up to these higher frequencies. with actually suppressed responses to low frequencies during broadband compared to sinusoidal motion, which is interesting because that's a phenomenon that's previously been described in central vestibular neurons. And so more recently, we've been inserting high density thin film flexible electrodes into the muscle in order to record a whole population of motor units while we're also recording neural populations in the cerebellum. And through our read-write probe, we can also stimulate neurons in the area that we're recording from. So for example, in nodulus uvula, when we stimulate, we can look at evoked responses in neck motor units, and we can also look at coherence between nodulus uvula Purkinje cells and motor units. So overall, just to summarize this talk, I've shown that anterior vermis Purkinje cells do these two key functions. One is transforming different streams of sensory input in order to generate appropriate commands to help stabilize head and body posture. They also provide the basis for distinguishing predictable versus unexpected self-motion. And in the RFN, during sensory prediction error, these neurons abruptly shift their encoding, even with very small deviations from the intended movement. So they have a very precise prediction of the sensory consequences of motor commands, despite motor variability. And the anterior vermis Purkinje cells, they show more gradual changes in their sensitivity, and the population activity of them is necessary in order to generate the responses in their target neurons. And finally, in the posterior vermis, the nodulus and uvula, these neurons combine vestibular and proprioceptive inputs in order to provide a vertical estimate or representation of head motion during both active and passive self-motion conditions. And overall, I think this body of work is crucial in demonstrating the powerful predictive capabilities of the cerebellum, which is essential for both enhancing the saliency of unexpected events, as well as updating our internal models in order to learn or in order to adjust over time to changes in our body and changes in the environment. So I want to thank funding sources, and I want to thank all of my lab mates in the Cullen lab, particularly my supervisor, Dr. Kathleen Cullen, and Omid Zobiri, who was involved in a lot of the work, especially in the anterior vermis, and Lex Gomez, who has been assisting me with the nodulus uvula recordings, Dale for his technical support, and then collaborators, Tim Harris, who's been highly involved in the work with the read-write probes, Faisal Karmali, who's been helping with some modeling with the nodulus uvula data, and Sylvia, who makes those beautiful thin film injectable EMG electrodes. Okay, you can take any questions if you're ready.
Thank you, Robyn. That was just wonderful. Yeah, we're open for questions, everyone. Please go ahead, and if you have any questions, let me know.
Reza, can I ask a question?
Yeah, of course, David. Go.
As far as the nodulus uvula, did you only rotate the monkey's head around a Earth vertical axis? Did you try any other axes where computation about gravity and linear motion might have to take place?
No, we have not yet. We have heavily considered putting them on a hexapod and doing that. Right now, I've just done linear translations, and we've started playing around with some tilts, some head-on body tilts, and also looking at potential changes in responses if you change the head-on body position and do translations. But no, I haven't systematically done any changes with changing orientation relative to gravity.
Thank you.
Can I ask a question?
Yes.
Yeah, thank you for this nice talk, and thank you for the nice surprise. I must say that to see the same responses during active and passive motion in the nodulus, this was unexpected, and this is going to change how we will think of it. But so the question is about the proprioceptive response in the nodulus. Are there some relation between like the translational response and the proprioceptive response? So if a Purkinje cell is activated by translation in one direction, is it activated by body motion in the same, or is there some rule?
Yeah, that's an interesting question because that also happens to be really different than the anterior vermis. So anterior vermis Purkinje cells, they tend to have like opposite phase responses to proprioceptive and vestibular input, such that they'll respond to vestibular input in one direction and proprioceptive input in the opposite direction, such that they tend to cancel each other. In nodulus uvula, that most often is not the case, where they show responses often in the same direction to both vestibular and proprioceptive, and then the combined response is almost like a sharpened response of the two other conditions. So it's more like they're combining that information to make a better estimate of head motion, perhaps, rather than using it to make an estimate of body motion, which is what the anterior vermis Purkinje cells are doing.
I see, thank you.
I have to ask raise a clinical issue. In the clinic, we tend to think that the patterns of postural instability are very different when the anterior vermis is involved versus the nodulus and uvula. And I wonder if you can relate the patterns to your findings of the difference in the way the Purkinje cells are responding. And specifically, classically, people who have anterior vermis lesions and alcoholic cerebellar degeneration as an example, they tend to have an oscillatory 3 hertz, 2 to 3 hertz oscillation back and forth. Whereas the patients who have nodulus lesions have a very different pattern of sway. They call it omnidirectional sway, and they just kind of will go off in any direction. And they're also worse when their eyes closed, as opposed to people with other lesions in other parts of the cerebellum. So bottom line, we have clinically very different patterns of postural instability between the front and back of the cerebellum. And I'm wondering if somehow the way information is being handled for posture is different, and if your work somehow sheds light on this clinical finding.
Yeah, thanks for that perspective. Actually, I think that fits really well with our findings, actually. And so, like you said, lesions in the anterior vermis, maybe especially with like a goal-directed movement, like a reach or something, you might see some tremor when there's some inability to make an accurate prediction of the sensory consequences of that movement, leading to some potentially prediction error that's maybe generating some tremor. Whereas also anterior vermis lesions have been shown to impair that ability to like distribute the vestibular spinal reflexes appropriately as you change head-on body posture, which is really important. The vestibular system is really clever. It only sends reflexes to muscles that are actually engaged in posture, which is what we see in human work. If somebody is holding onto a railing, you'll see reflexes in those arm muscles, but not if that arm is not engaged in helping support the person. So it seems like the anterior vermis is crucial in that, whereas the posterior vermis likely has more of that role of integrating canal and otolith and proprioceptive information as well in order to stabilize things depending on our orientation with respect to gravity. So if there's maybe some impaired estimation of how the head is positioned or moving relative to gravity, maybe that's where you see those impairments in the postural responses.
Yeah, okay, thanks.
Okay, if I may ask another.
Sure, go ahead.
So regarding the anterior vermis and the fastigial nucleus and the cancellation. So through, I think, all this year, there's been clearly an understanding that you have a sensory signal coming from the vestibular organ and you have a cancellation signal cancelling it during active motion. And then it feels like what you were expecting is that the Purkinje cell encode this cancellation signal and then that the fastigial nuclei receive both the vestibular signal and the cancellation signal, which would imply probably that MOSI fiber to the fastigial nuclei would encode that cancellation signal. Now, is it indeed, how do you see it? And then considering that after all now you see that Purkinje cell in the anterior vermis are inhibited during active motion, do you see maybe another possibility, which is that the fastigial nuclei are carrying a copy of Purkinje cell activity?
The vestigial nuclei might be carrying a copy of the motor.
Yeah, I don't know if it was clear. So once again, to put it a little bit simpler, is it that the Purkinje cell encode a cancellation signal and then the fastigial nuclei combine this cancellation signal and the vestibular afferent signal? Or is it that the computation is made upstream?
Well, based on the population modeling that Omid did, it is possible that the cancellation signal is generated purely by the Purkinje cells. It doesn't mean that might be other mechanisms of doing it, but it is fairly intriguing that a very reasonable number of Purkinje cells can predict responses during active conditions. And it's not as simple as this pure cancellation signal coming in on the Purkinje cells. It is more of pooling their heterogeneous responses to both the sensory information from the self motion and the motor command information. And we think that might be valuable because when you have these sort of heterogeneous responses, it's easier in terms of learning to sort of adjust the weighting of these different Purkinje cells in order to generate like appropriate responses as for example, your body changes or things change in the environment, anything that requires some updating or motor learning. So based on that work, it is possible that reafference cancellation is generated by the Purkinje cells and might actually make sense in terms of, if you look at it in terms of the role of the cerebellum in learning.
Yeah, I see. Thank you.
Thank you, Robyn. Are there any other questions for Robyn? All right. Thank you, everyone. If there are no further questions, I will see you next week. Good to have you with us.
Thanks,
Thank you,
Reza.
Robyn.
Thank you.
Bye.